Well, welcome to the final plenary session of the meeting. We have a really exciting program for you this morning on standardization and MR. But before we get started, let me introduce myself. I'm Lawrence Wald, Larry Wald, and it's an honor and a pleasure for me to be able to serve as the president of this society. Today is my first day. And I clearly remember my first meeting in the early 90s and the feeling that this was going to be my professional home here at this ISMRM meeting. And since then, it's grown to include this amazing, broad group of mentors, colleagues, and personal friends. And it's nothing but a pleasure to be able to serve this group as president. Thank you. So, thank you, and I'm definitely looking forward to a fun and productive year. So now it's my pleasure to announce a final series of awards for the program. And I'm going to start with a really fun one, the Fun Run winners. And we have this remarkable picture of two very athletic-looking members, John Herford and Abigail Sember, who won our 2019 ISMRM Fun Run. Big round of applause. Then the next item on the agenda, I guess I have the slides, is the ISMRM Research and Exchange Grants. These grants were designated by the ISMRM to foster the exchange of ideas and the advancement of basic science, preclinical and clinical magnetic resonance imaging and spectroscopy research, by supporting the exchange of researchers to alternate research sites. And just an FYI, we'll be looking for the next round of proposals in February 2020. So this year's three winners of these exchange grants were Sanjeev Chawla from the University of Pennsylvania, and he'll be investigating the potential of multi-parametric MRI using machine learning in redefining treatment response in patients with glioblastoma. A round of applause for Sanjeev. The second recipient was Klaus Eichel from the Fraunhofer Mavis Institute in Bremen, and he will work on image reconstruction of simultaneous multi-slice data at ultra-high field without scan-specific received coil sensitivity collaborations. And let's give him a round. And then the third award goes to Hanna Sechsten from the University Medical Center of Göttingen, and she will evaluate the association between enlarged perivascular spaces as visible on magnetic resonance imaging and subsequent rates of hippocampal atrophy in subjects at risk for familiar and sporadic Alzheimer's disease. So congratulations to Hannah. And then it's my pleasure to announce the 2019 Junior Fellow Symposium Challenge winners. So as just a little bit of an explanation, the Junior Fellows organized this event, and they defined specific challenges specific to different parts of the world. They had a competition whereby proposals were sent in into how to meet these very difficult challenges, as you can see. And we had six challenges that won this competition. And hopefully the next step will be to foster collaborative efforts and pursue these challenges. The Africa challenges were a virtual scanner software by Yihao Tong, and all you need is a browser eliminating barriers to MR education with open source interactive tutorials by Matthew Boudreau. So congratulations to them. The South American Challenge winners were a web-based data management and processing system to support prevention of vascular disease vascular dementia by Norman Young, and the prevention of vascular dementia by Sina Straub. And finally, the two winners of the India Challenge were on generative adversarial network-driven synthetic MRI by Adharsh Ghosh and a concept of screening MR for stroke in India by Sunita Gudwani. So congratulations to all of our Junior Fellow Challenge winners. So now it's my pleasure to hand the podium over to our new Vice President of the ISMRM, Tim Leiner, who will be presenting the Young Investigator Awards. Tim?
Thank you, Larry, and thanks for coming to this important awards ceremony. So it is my great honor and pleasure to announce the Young Investigators Awards. And as you may know, there are two awards. The W.S. Moore Awards is focused on rewarding original clinical research in MRI. And the Robbie Award is focused on original basic science research in magnetic resonance imaging. And the candidates for this award are clinical or basic scientists at the undergraduate, graduate, or postgraduate level for basic scientists, and at the resident or fellow levels for more clinically oriented research. So within 5 years of getting their PhD, or within 3 years of completion of clinical training. And this is a very rigorous process, the selection of the finalists. So being nominated is already a great achievement. And the candidates send in a manuscript, which is then reviewed by the committee chaired by Harry Hu. The authors are then required to present an abstract and a presentation at the meeting, and they are interviewed by the committee. So this is already somewhat of a marathon for the people who are actually undergoing this process. So let's look who the finalists are. And for the Robbie Award, this is Matthias Davids from Heidelberg University, who presented a paper on prediction of peripheral nerve stimulation thresholds of MRI gradient coils using coupled electromagnetic and neurodynamic simulations. Carol Lazarus from Neurospin with a paper on sparkling variable density K-space filling curves for accelerated MRI. And Fuji Shui Yuang from the Atinula Matinos Center for Biomedical Imaging, and with a paper on echoplanar time-resolved imaging. So if we can have a drum roll, I can invite the II Raby Award winner. Of course, this was a very difficult process, as I explained. The candidates were all very good and had excellent papers and presentations. But it is my great pleasure to ask Matthias Davids to come to the podium. Okay, if we can go back to the previous slide, we will talk about the Moore finalists. So the first finalist is Tess Armstrong from the University of California, Los Angeles, and she presented a paper about 3D R2 star mapping of the placenta during early gestation using free-breathing multi-echo stack of radial MRI at 3 Tesla. The second candidate is Akshay Chowdhury from Stanford University with a 5-minute double echo in steady state with separated echoes for comprehensive whole joint knee MRI assessment with and without a proton density weighted sequence. Definitely wins the prize for the longest title. And then we have Angéline Nemet from the University of Lyon with 3D chemical shift encoded MRI for volume and composition quantification of abdominal adipose tissue during an overfeeding protocol in healthy volunteers. So if we can have a drum roll again, I would like to announce the winner of the W.S. Moore Award, which is Akshay Chowdhury. So this was the final award, and now it's my pleasure to invite the organizers of the plenary up onto the podium, Dr. Mustafa Bashir and Dr. Chiong Gong.
Thank you, Tim. Thank you, Tim. Good morning, everyone. Thank you for joining us for the last plenary of the meeting. I hope it's been a fantastic meeting for everyone. I'm Mustafa Bashir from Duke University, and my co-moderator is Dr. Chiang Gong from the West China Hospital at Sichuan University. And the plenary this morning is entitled Standardization in MRI, Why is it so challenging? And the idea is that no matter how you interact with MR data, whether it's on the acquisition side, the processing, or the interpretation, we rely on certain assumptions and certain standards in order to help us understand the data and to help clinicians understand our interpretations. And these standards can be very difficult to come by. They can be very difficult to understand. And from the outside looking in, you might ask yourself, why is this? And what can we do to enforce more standardization to help us and to ultimately help our clinicians take care of patients? So we have a fantastic line of speakers for you this morning. I'd like to first introduce Dr. Mitch Schnall, who is a professor of radiology and the chairman at the University of Pennsylvania. He's also a group co-chair at ECOG Akron and has a tremendous amount of experience distributing imaging biomarkers in multi-site clinical trials. And we're looking forward to his perspective. Please welcome Dr. Schnall.
So thank you. I wonder if I can have the first slide. So I really appreciate the opportunity to come and share some of my perspectives on this question of why is standardization so challenging, really focusing on machines and acquisition protocols. And I do this as was just mentioned, from the perspective of someone who's wrestled with trying to bring the cats together to run multi-center clinical trials, and also within the department to bring the cats together to establish clinical protocols and what that's like. And as a little bit of a spoiler alert, you know, a large part of the problem relates to culture and not necessarily to technology as we'll go forward. Now, another disclosure is we've got a grant from Siemens, mixed disclosure. So, you know, I've been doing MRI for a long time, and over that time I've seen astonishing developments in this field that have positioned MRI to make unbelievable impact both as a clinical instrument and as a research instrument. And I think when I think about it, there are several things that have driven this, but I like to think of three major drivers of what we've seen in the advances of MR. One is the richness of the MR physics that gives us a great playground to play in. The second is unbelievable creativity, frankly, by the community represented here to leverage the richness of the MR physics in order to develop, you know, unique methods that continue to advance our specialty. And the third is a fierce competition over brand, and that's a fierce competition both in the industry side as well as on the academic laboratory side and individual clinical practitioner side in terms of establishing your methods, promoting your methods, and trying to establish that as essentially a brand. And so these things working together, I think, have driven incredible advances in our field. When I think of what the challenges are to standardization, the same thing. The richness of MR physics gives us a huge menu of options to choose from. The creativity of the research community has taken advantage of that to create really unique opportunities to to study individual disease processes and individual organ systems. And of course, the fierce competition, we promote the innovation, we promote things that we do unique. Everybody wants to be the first one to show the best pictures in the next meeting. And so those are the things that sort of drive us, if you will, into individual approaches to practice and really impede our ability to come together into harmonized protocols. And so with that, we can pretty much finish, but I'll dig into it a little bit more. If we think about the richness of the MR signal, this is just a subset of some of the basic methods that are available clinically today. I could probably put about 2 or 3 slides like this up with a lot of methods and probably add about 4 or 5 from what was done at this meeting today, so this continues to grow. So we've got a large menu of options in order to attack our disease sites that we want to study. Even if we look at a single sequence, a single approach, and look at the flexibility within that approach, let's look at the degrees of freedom within a single gradient echo pulse sequence, which arguably is our simplest pulse sequence. It's a single pulse. It's a single echo that we acquire after a TE. It's awaiting a TR period and taking another echo. We have variabilities in field strength. We can decide whether we want to refocus transverse magnetization or not. We have TR, TE, flip angle, the standard sort of geometric issues of matrix field of view slice thickness. We've got bandwidth. We can decide what we want to do with water and fat. We can look at K-space trajectory and parallel imaging, which are interrelated. We have RF pulse shapes, gradient waveforms, coils, and more. And so there's a lot of opportunities to tweak acquisitions as we go forward, which again feeds into the creativity of the field to continue to evolve very specialized approaches to how we study disease. And these can have, fairly significant impacts. So these are 4 gradient echoes through an abdomen. If we look at the top two scans, this is just a subtle change of about 1.5 milliseconds of chemical shift evolution, and we go from fat and water being in phase to fat and water being out of phase. Most of you have seen this before. Makes a dramatic difference in the image even with a subtle change in the acquisition. We can add a preparation pulse to get rid of fat. Again, dramatic changes in the appearance of the image. And we can decide we're going to refocus all the transverse magnetization and have a steady state free precession component of the signal and a very different looking image. And I'm sure, by the way, all of you saw all these little dots in the liver which represent multifocal hemangiomas, just to throw a little clinical aspect into there. We can also do things where the images look pretty similar, but we go at it in very different ways. So these are 2 images. The one to the left is a standard rectilinear encoded 3D spoiled gradient echo. You can see an enhancing lesion in the breast for those that don't do a lot of breast MR. And here's a fairly similar looking image done in a very different way. This is a radial acquired image using golden angle increments, using a quick time filtered reconstruction. So this is about a 2-minute acquisition. This is about nominally 15 seconds acquisition, or 15-second time resolution. Now, I got to tell you, when we unleashed this in about 2004, we were really proud. I went over the country talking about breast MR, exciting to show what I was able to do, proud to show these 15-second video clips at high resolution of contrast going in and out of the breast. And that gets us to this next issue, and that is the idea of fierce competition over brand. And I want to try to make this a little real and give you some context here. So back in the early 2000s in breast MR, there were effectively 2 major brands of breast MR, right? A lot of folks were doing it, as illustrated to the left, dual breast interleaf sagittal acquisitions. There were other folks that were doing it as an axial or sometimes coronal acquisition, having both breasts in the same field of view. And in fact, there were almost like religious cults in these 2 domains. There was fierce discussion over which is better. People who practiced one way wouldn't consider even thinking about practicing the other way. And of course, we know now that frankly it doesn't matter, that the clinical results are the same either way. And in fact, we've switched how we do it at Penn. And to tell you how fierce this is, we did a clinical trial, and this is our Akron 6657, which is a neoadjuvant therapy clinical trial. And to do this, we tried to require very, very tight control of protocol. And so here was the correct protocol was a sagittally acquired image. You can see the protocol, defined sagittal, defined, very tightly this protocol. And here's what a number of sites actually submitted. They refused to compromise their brand. They couldn't practice the other religion. They had to stay with their brand. And that's how fierce this allegiance to brand is. And often this brand includes an alliance between an industry manufacturer and a thought leader in the field that get together to define that brand. They promote it together, and it, in many ways, divides the specialty and makes it hard for us to come to harmonization. Now let's think a little bit about the benefits of standardization. Really, it's about, as we just heard, creating data sets that we can leverage. We can leverage for research. We can leverage for training AI algorithms. God knows we've seen enough about AI algorithms over this week, and potentially reduce the variability of clinical practice. Does it work? Sure. I would bet that 3/4 of this room knows about the ADNI consortium. ADNI consortium under research protocol collected under 3 different protocols, ADNI 1, 2, and 3, had collections of relatively homogeneous images. They've made them available. You can see all the downloads there were, over 1,700 publications, and a lot of progress in biomarkers of Alzheimer's disease because of this data set that is consistent. So consistent data sets, the value is really there. I'd argue that in thinking of standardization, we're thinking of it a little bit wrong. We're thinking of it from a process conceptual flow. In fact, the organization of this session is in a process conceptual flow. We think, how do we standardize the machines? How do we standardize the data processing? And then what does that mean for interpretation? What we ought to be thinking about it is the other way. What drives it? If we're going to impact this, we've got to think about what drives it. And if we look at how decisions are made on the ground in protocols, and we'll talk about that in a minute, it starts with what's the clinical need. It starts with how do I interpret the images to drive the decisions for the clinical need, and then what governors do I have to put on machines and protocols so I can do the interpretation that I need to do to drive the clinical care of the patient. So we have to think about the drivers and not think about it from a process perspective. If we think about clinical standardization, there are a number of groups that are trying to standardize aspects of our practice, and probably the one that has been most successful, most impactful has been the American College of Radiology. The American College of Radiology, through its BI-RADS, LI-RADS, PI-RADS, I saw a session here on PI-RADS, has been slowly going and trying to rein in and standardize aspects of clinical practice. And it starts with standard reporting based on clinical need. That standard reporting drives some exam protocol requirements, allows us to benchmark clinical performance, establish effectiveness. And the one positive is some of this can be enforced through clinical accreditation programs. So people sort of have to meet these protocol requirements because of the clinical accreditation programs. But remember, the protocol requirements are not driven by what's possible. They're not driven by research interest downstream. They're driven by clinical need. And so if you look at these lexicons, what they tend to do, and I realize this is the actual lexicon downloaded, so I wanted to show you the entire lexicon. But let's look at one aspect of it when they talk about the kinetic aspect of interpreting a breast MR, and this is the breast MR lexicon. They divide the interpretation into an initial phase and a delayed phase. And you're asked to choose whether it's slow, medium, or fast, or whether it's washout or persistent or plateau. And so they drive the clinical protocol requirements based on that need, right? So if you look at the clinical protocol requirements, and this is right out of the accreditation manual, pre-contrast T1, early phase, delayed phase, says it's got to be completed within 4 minutes. Why is that? Because they define the early phase as 2 minutes and figure in rectilinear case-based sampling, you're kind of in the middle if you do continuous traditional sampling at about 2 minutes. And they also give a little bit of guidance, with 3mm slices and 1mm in plane resolution. So these are protocol requirements which, in fact, you have to meet to be accredited to do breast MR through the American College of Radiology, so they can be impactful. Similar requirements under PI-RADS. PI-RADS, like a lot of the standardization of clinical practice, assigns scores which meet. Each of these scores is assigned to a given diagnostic category, which means a given clinical recommendation. And you can see these scores are driven by the DWI score, most important in prostate MRI. And so the DWI protocol suggestions are made very clearly in the lexicon. And so if we can influence the clinical practice standards, we can influence the standardization of protocols. So how are protocols on the ground put together? Protocols are usually put together through things like protocol meetings. This is a protocol meeting at our place at Penn. This is our abdominal imaging group in a protocol meeting. We've got represented here a group of our physicians that practice body MRI. We've got a number of technologists and technologist leadership that's involved in MRI, and they get together and they all bring their biases to this protocol meeting. They bring the biases of their experience, biases of their brands, some of whom have promoted brands locally, some of whom have national prominence and have promoted their brands across the country. They bring the history of that practice at the institution, and they need to put them all together within the capabilities of the instruments and economic factors in order to develop protocols. And these can be rather contentious discussions, I can assure you. And so all these things are going to go into defining protocols. It starts with applicable standards. And as I said, if we can influence the standards, these are paid attention to in developing protocols. Clinical need, individual radiologist preference is important. These radiologists are interested in how do I get through my day? How do I effectively take care of patients? How do I mitigate any risk that I'm going to be missing something that I should be taking care of? Physicians are very, very concerned about their patients. There's referring physician preference. Occasionally we have referring physicians that say, I need to see it this way because this helps me understand how I'm going to operate on this patient, or this helps me understand, how I'm going to talk to the patient. There's instrument capability. There's economics, the economic reality of continuing decreasing reimbursement, certainly in the States. And yes, people do consider value of data and research, but it's considered within quite a bit of noise of a lot of other considerations that need to be thought about in developing protocols. And so again, I'm pointing at applicable standards. That's an area that we can potentially influence through some national organizations. So I want to speak just for a minute on quantitation. A lot is talked about quantitation in MRI. I don't know how many people know about QIBA, with Quantitative Imaging Biomarkers Alliance. It's a program that had been funded by NIBIB and housed at RSNA. And it looks at imaging and trying to develop quantitative standards for imaging that manufacturers and practitioners could adhere to. Problem is that it's driven primarily not by clinical need. It's driven by people in the clinical trial process. Now, I don't know about you, but we do, within Penn Medicine, we do about 500 MRIs a day. I'd say arguably 480 of them are done for clinical goals, and about 20 of them on a good day are done for research purposes. But there's some unique problems quantifying MR. In addition to all the other things that imaging has, which is what our reconstructions do and partial volume averaging and all that, we've got problems that a lot of our parameters are affected by field strength. We've got a lot of B0 heterogeneity, which is different for every patient based on how they interact with the B0 field. We got B1 heterogeneity based on how we load coils, and in fact, do we ever really know what flip angles we're going to give in what part of the patient? We got receiver coil sensitivity profiles. And so really what that pushes us to is to think about low-hanging fruit for quantitation. The low-hanging fruit are areas that are gradient-based, because we usually know what our gradients are going to do. They're pretty reliable. And where there's an internal reference. And so where do we have that? Measuring motion. We're pretty good at measuring motion, and that we can do relatively reliable. What are more important motion measurements that are important clinically? Diffusion, right? Flow, and I'd argue elastography is really just measuring motion in a unique context. And so these things, I think, we can actually measure. QIBA has actually been very active in DWI, developing a profile for DWI just for measuring the ADC. They define the best practices. In other words, how should you QC a scanner? How should the scanner be set up? They talk about protocols and start to summarize what's known about reproducibility. And so they've done a whole profile in DWI, including prostate. And what they'll tell you is that the expected coefficient for variance for the ADC of a prostate tumor is about 17%. Now, before people get upset, that's not the ability to measure a DWI, right? An ADC. If you take a water bath phantom, send it around, you can measure ADC within 3%, right? But you put a lesion in a patient with the issues of biologic variability, with the issues of heterogeneity and sampling errors, with the issues of partial volume averaging, you're going to be within 17%. And that clinically significant change in ADC, estimated to be 47%, pretty significant, and that's for our low-hanging fruit. So there's still some work to be done there, and the question is how much of this is biologic variability versus machine variability. As I said, you give me a phantom, we can measure within 3%. One of the challenges we have with a lot of these analytic approaches is breaking into this cycle. You want to get protocol adoption. To do that, you've got to demonstrate value. How do you demonstrate value if there isn't protocol adoption? The best way to break in there is through clinical trials. Clinical trials could be an aggregation of small trials that are all done similarly and point to the same direction, or a larger multi-center clinical trial. If you do a larger multi-center clinical trial, generally the biggest issue is how are we going to get support to do it, and how are we going to disseminate the technique and support the technique across multiple institutions, which is a challenge. So I just want to conclude by pointing out that, frankly, many of the drivers of our success as a community are the same things that cause challenges to standardization. So we've got to think about that very carefully when we think about our zeal for standardization. That a lot of things influence our protocols. Applicable standards, radiologist preference, ease of implementations, economics. The one that we can influence, potentially, is the applicable standards, and to create evidence to show that certain protocols are preferred in taking care of patients. And then finally, as I said in the start, frankly, this is more of a cultural problem than a technical problem. There are certainly technical problems, and I am certain that this community is capable of solving most of the technical problems that are out there. The bigger challenge is the cultural problem that, frankly, as a community, we champion the uniqueness of what we do. We champion the innovations that we create as opposed to standardizations that we could potentially achieve. Thank you. Take questions.
So thank you, Dr. Shino. It's a great pleasure for me to introduce our next speaker, Dr. Vince Colhoun from Man Research Network, New Mexico. Dr. Colhoun has a tremendous amount of expertise in terms of image analysis for the multimodal imaging, which contribute to the clinical translation of neuroimaging, such as psycho-radiology. So today, Dr. Colhoun are going to talk about the standardization of data format and the data post-processing method. So please welcome Dr. Colhoun.
Thanks. It's a pleasure to be here. What I was going to do in my talk is focus mostly on issues dealing with side effects in research studies that we've encountered for brain imaging. But before I go there, I wanted to just touch on a few efforts in the field to get towards standards for post-processed data. First one is brain imaging data structure, which is really a recognition that that we need more than just an image to do the study. So we need imaging data, behavioral data, sometimes multimodal data. We need to capture information about the derived information, and it all should be in a place that's standardized that can help us to replicate results. And so that's one effort. Another effort is to try to standardize statistical models. And so this one example is the neuroimaging data model, which is spearheaded by the International Neuroinformatics Coordination Facility. And this is really an attempt to try to standardize statistical results coming out of different packages. For example, FSL and SPM doing a general linear model have lots of different parameters, lots of different ways of storing the data. And so this tries to standardize that, capture information about provenance, and enable us to be able to do comparisons more easily. And then thirdly is the need for more sort of society-led white papers and information about how do we report results in research. This is an example for the Committee on Best Practices in Data Analysis and Sharing, which is really focused on MRI-based measures in brain imaging, and really tries to document, again, how do we report what we've done? What are the different parameters that we should be looking at, et cetera. There's a new version that just came out also looking at MEG and EEG. So now let me just talk a little bit about site effects or scanner effects in research studies. So one way to kind of visualize this is to compute quality control metrics. So this is an fMRI study And here I'm just essentially clustering quality control metrics. So this is a two-dimensional plot. It's called T-stochastic nonlinear embedding, where it's essentially just a two-dimensional cluster. Each dot on here is an individual data set. And you can see there's sort of 4 big clusters here. This one here is sort of, you know, places where the processing has gone wrong. Over here, we've got individuals that have a lot of motion associated with them in an fMRI study, and then the bulk of the data is here. So that gives you kind of a high-level perspective on what's going on with any individual data set, but it can be depressing at times. This is an example of a retrospective analysis from the ABIDE brain imaging data exchange, and you can see that if you just cluster on QC metrics, almost every site gets its own cluster, right? So that's not what we want. And you know, thankfully, the news is not that abysmal. If we look at, once we deal with the quality control, then start to look at the information that we actually want to find that's consistent across the site. So here's an example of basically processing T1 images into gray matter segmentation maps, and then doing a voxel-wise analysis of those maps. And what I'm showing you here is 10,000 individual data sets on 3 different scanners, 2 3T and 1 1.5. And I've color-coded these by field strength here. So the bad news is that they don't overlap perfectly. But the good news is, if you look on the right here, if we color-code the same plot by age, from young to old, you see essentially the same pattern in the 2. These are infants over here that get their own cluster. Suggesting that, you know, it's not all that bad, and there's some, you know, relatively simple things we might think of to sort of calibrate this. Another way that, so one of the ways that we try to deal with sites is you can co-vary by, you know, site effects. You can also basically record all the parameters that were used and try to use that to clean up the data, at least to remove those effects that are associated with those parameter differences. So here's an approach based on independent component analysis, where we essentially take, again, these voxel-wise gray matter maps. So we have a voxel-wise data and then subjects, and then we're basically extracting components that are co-varying. They're showing gray matter patterns that co-vary across these individuals. And then we do a regression. We fit all of the different parameters to those components and identify the ones that are strongly associated with them, and then remove those from the data. So here's an example from 2 large multi-site studies. I know it's hard to see, but they're basically a lot of parameter differences between, in this case, 4 sites, and this is actually a smaller number of sites, but it was many, many years, about 8 years of data collection or more, lots of parameter changes over time. The components that were identified as most associated with those parameters are down below here. And you notice they're mostly limited to components that are sort of edges in the brain, in some cases the potentially susceptibility in this inferior temporal lobe region here. But then once we identify these and we remove them, we look at group effects, in this case schizophrenia versus healthy control. So I'm going from top to bottom here, where I've got essentially a component that shows a significant group difference. Uncorrected, this is a voxel-wise regression correction, and this is a component-level correction. So the good news is that they all look kind of similar. The components that were removed in the correction had strong effects associated with field strength, associated with resolution, and various parameters, as you'd expect. And the good news is that the variance explained in terms of the group effect actually increases by about 6% going from top to bottom here. So that's just one approach. Another approach is to, if you're doing a study, you know, have a traveling phantom, you get a human and you scan them at all of the sites, use that to do some sort of a calibration. Here's an example of a coefficient of variation for within subject, cross-site, and between subject for cerebral blood flow measure. We can also look at some of the other measures, for example, gray matter and white matter for each of the 4 sites. They're pretty solid. We've got flare effects, T2 star maps. The functional measures are down here. Those are a little bit more variable, but that's to be expected in some sense because it's a functional measure. But you can see across site, it's fairly, the means are sort of consistently in the same direction. Here's an example from diffusion MRI where we scanned a few individuals on 2 Siemens scanners. And then what I'm showing you here is for different tracks, sort of fractional anisotropy within those tracks. It does follow pretty closely. We get an average error about 3%. If we scan Siemens versus GE, we get about a 9% error, so the inter-vendor differences are greater than within vendor. And then if we use just a simple calibration, sort of a ratio-based correction of one individual, and then use that to look at how the effects change on a different individual scanned at both scanners, we go from about 6% difference to about 2% difference. So that actually, those calibration techniques actually help, it appears, to remove some of the differences associated with just the scanner. Now, if we look at a more larger scale study, this is the Enigma Worldwide Consortia. Just a couple examples from that, where basically it's a consortium of studies, and analyses are set up and scripts are run locally, and then they're passed to a central site where they do a meta-analysis. And so this is looking at schizophrenia effects on hippocampal volume. You can see here the good news is that almost everybody is to the left, actually everyone is to the left of the line in terms of the mean. There's some variation here, of course, but in general, this is good news. And here on the right is another study looking at negative symptoms. And again, with the exception of this one that falls right on zero, every other site is to the left, showing a negative effect. And so this is an example where we're controlling for site because each study had both cases and controls, and so they're calibrating out site that way. Another example where we look at sort of the ranked group differences in about 5,000 individuals from largest to smallest, or reversed, and then we compare in an independent large study and do the same sort of ranking. Look at how similar this looks in terms of the ranking. We get very, very similar effects for hippocampus volume changes in patients and controls in an independent study. So this is good news. Does this hold also for resting fMRI? So a connectome-based approach. So there are a lot of ways to sort of parcellate the brain. We tend to focus more on data-driven approaches, so like independent component analysis. But those approaches can be challenging to generalize because you have to select components at each individual analysis, et cetera. So the way we've dealt with this is we run a group analysis on independent normative data sets. So here we've got Human Connectome Project and Genomic Superstruct data. Then we do an intersection of the components that are consistently identified at both to generate some templates here, which are then fed into a single subject ICA as a prior, spatial priors. So the advantage of this is that you don't have to select components. They're already ordered, they're already structured. It also allows adaptivity. So in contrast to a region of interest-based approach, it adapts to individual variability in subjects. So how does this work? If we look at the connectivity between components here for two independent data sets, so these are separate from the ones that were used to derive the templates, you can see that we're getting pretty rock solid maps. They look very similar, correlated above 0.95 for main effects. The effects we usually care about are these small effects. So here we're looking again at schizophrenia and healthy control. And you can see on the whole, the patterns do look pretty similar. We get, for example, this block here between cerebellum and sensory motor showing up in again, these are 3 independent data sets here. If we threshold the most significant differences and look at the intersection, we're again above about 0.95. So how does this work in practice? This is an example of a study where we were looking at predicting something that has some potential clinical relevance, so looking at medication class response to either antidepressives or mood stabilizers in euthymic individuals that were young. And so they get a scan at first presentation, and then they're followed up for about 2 years to eventually determine which medication they show a positive response to. And so that's in a sense, our gold standard in this. And so we applied that connectome-based approach, and we find that we can predict with about 90–92% accuracy which medication the individual will respond to, even for complex patients that were unable to be diagnosed in the clinic initially. And the other good news is that it also tells us information. We can identify which brain networks are most informative in that regard. So it gives us localization. It tells us information that could potentially be used for identifying candidate targets for treatment, et cetera. Dynamic connectivity is really kind of an extension of that, which is looking at changes over time in these correlation patterns in resting fMRI, for example. And so the question really is, well, a lot of studies have shown that this kind of an approach is more sensitive if we're looking at prediction and classification, because we're capturing that variability that's occurring within the data. But how robust is this to looking at sort of scanner site effects? So this is a replication study we did just using sort of local scanners. So it's fairly harmonized, but we have 2 3Ts and a 1.5T in this study. We had 7,000 data sets, and we split them into 28 independent analyses and looked at how similar the results were across the different analyses. And we find that the patterns for the different transient connectivity patterns were correlated above 0.8. We also find that individual dwell time, so if you tend to spend more time in a particular pattern, that was consistent across those independent analyses. Again, correlated above 0.8. And here's just a distribution of some of the explicitly dynamic parameters. For example, total distance traveled in the state space and total number of states entered, for example. And you can see there's variability, but they're pretty stable, pretty central, and this is after covarying and trying to remove motion effects from the data. So yet another extension, and one of the real strengths of MRI is that it's highly multimodal. As the previous speaker mentioned, we've got a lot of different choices, a lot of parameters. That's also a strength in addition to a limitation. And so, there are a lot, that sort of screams out for approaches that can analytically combine the data fusion approaches. So I'm not going to go into algorithms, but just to mention that, one of the ways to do that is to look for commonly covarying patterns in a given individual across, for example, brain structure and brain function. And so we're using, some techniques based on ICA to do that. And so here's an example from a large study where we basically have 4 different modalities or features, so to speak. We have 2 fMRI measures, regional homogeneity and functional network connectivity from ICA. We have a structural MRI, which is gray matter, and we have a fractional anisotropy. And you see we get significant, again, this is a patient versus control schizophrenia comparison, increases in REHO, decreases in the other 3 measures. But the interesting thing is this is after we covary for site. But if we then divide this into Siemens and GE, for example, notice that we get actually the same effects. Really, the directionality is consistent across these different scanners for all 4 of those modalities. Fairly similar patterns in terms of the most dominant connectivity patterns and changes. And this is also true for a continuous measure, looking, for example, at working memory. So we get working memory effects that are, again, consistent. This is, again, the Siemens data. This is the GE data here. And then the last example of data fusion would be here, suppose we look at something like cognitive scores. We're not completely blindly decomposing the data. We're actually telling the algorithm we want to find joint features that are maximally associated with a cognitive score. Right? So that's sort of a constrained approach. We have multimodal data. We extract patterns that come out of that analysis that are associated with cognitive scores, find consistent regions among them, and then use that to develop predictors that we can use to classify different data sets, new data sets, et cetera. And here I'm showing you results for cognitive score versus modality weight loading parameter for 3 independent cohorts based on that prediction. And you see very similar sort of patients to the right. They tend to have higher loading parameters, controls have higher loading parameters and higher cognitive scores than patients. And so this is actually looking quite encouraging. So another real important question is, as algorithms get more and more sophisticated, they're going to be able to cheat more too, right? So they're going to look at anything they can to try to optimize. And so I think that's the tricky part here is, for example, deep learning methods, that this is applied to the ADNI data, looking at a deep residual network. But are they going to be able to, in a sense, inflate the correlation based on side effects in some way that we aren't really able to determine? So here is just showing that, we do, like many, get a high accuracy in predicting Alzheimer's versus healthy controls, over 90%, about 75% in predicting who's going to convert from MCI to Alzheimer's, and these are the regions that are sort of most informative there. But this is just sort of a teaser, which is, suppose we regress out site prior to doing the deep learning model, and then I'm running sort of 7 folds of the data, and you see, except for in this one exception, in most cases the performance is decreasing. So is that good or is that bad? What is that telling us? Is it leveraging the side effects to improve performance in some way that's sort of cheating? Or is it telling us that regression is removing useful information that maybe is unrelated to sight? Answering those questions is going to be very important going forward. I don't have an answer for you yet, but we are running some additional studies to try to look at that using sort of different ways of cross-validation, different ways of visualization, for example, relevance propagation, to see what the regions involved look like to help us answer those questions. And in just the last few seconds, I just wanted to mention that there's actually, I'm going to skip this section because I don't have enough time. I will summarize that, I think we're still learning how to best address side effects. We can use, parameter details combined with machine learning to help identify those and remove them. We can covary or calibrate out side effects as best possible. Interestingly, pattern-based approaches tend to be robust to side effects. So if you design a pattern-based approach where you're looking at sort of relationships between regions, that tends to give you good results regardless of site. So that's also interesting. It's a question of how big these effects are and how much they impact the results. And an extension of that is multimodal data fusion, where the relationship between modalities appears to give us an additional beneficial lever to help identify and remove side effects. And in summary, I'll say that there is a lot more work to do, and maybe I've raised more questions than answers, but hopefully this has been helpful. So.
Thank you very much. Thank you very much, Dr. Kohong. It's elegant work. Well, in sense of time, I have to move on to the next speaker. Next one, Dr. Lisbeth Renneman. Dr. Renneman is a professor of radiology from the University of Amsterdam. She has a great deal of experience in translational neuroimaging studies, and this which contributed a lot to what we say new field of psycho-radiology. She's going to talk about the standardization MRI and why it's so challenging, specifically about from the psycho-radiology perspective. So please, welcome.
Thank you very much. I thank the organizers for the invitation for me to talk about standardization of clinical interpretations, the psycho-radiology perspective. I have nothing to declare.
Do you happen to know who the purchaser was?
Why, yes. He left his card, just in case anyone should inquire for him.
How interesting. I'm sorry, but I'm afraid I have to look a bit further. Thank you anyway.
Good afternoon, thank you.
People put the strangest function titles on their business cards. Mine says psycho-radiologist. It's a function that didn't exist until about 2 years ago. But we need more of them, rapidly. Let me tell you why. Every year, 1/5 of our population suffers from a mental health disorder. I'm not sure if this audience is very representative for the rest of the population. But if it would be, it would mean that in this room alone, about 1,000 people are suffering from a mental health disorder. And more than 1.5 billion people around the world. So what kind of disorders are we talking about? Some of the most frequent ones are anxiety, depression, insomnia, alcohol and drug dependence, ADHD, mostly among the young, and dementia, mostly among the old. Many people suffering from a mental health disorder receive medication. And now we're getting to the real problem. Our medication is not working. Or, to be more precise, our medication is not working good enough. And let's be clear, few medications always work. A meta-analysis of the 20 most common drug types found that most demonstrate a minimal clinically important difference. the biggest effect was found for proton pump inhibitors for reflux esophagitis. But the efficacy of medications for mental health disorders is particularly depressing. Often, people receive psychotropic treatment for which any evidence is lacking. It's a trial and error approach at best. And for some groups, particularly children, I would argue that the evidence based upon which we give them medications is rather reckless. The diversity of available drugs shows the difficulty of finding the right drug for the right patient. For schizophrenia, for instance, there are over 30 suggested drugs. About a third will have partial response, and of those responders, a third will relapse. For depression, a third to half of all patients will have full remission with treatment. Most patients receive selective serotonin reuptake inhibitors, also called SSRIs. Other drugs include, and I'm getting to the tongue twisters here, norepinephrine dopamine reuptake inhibitors and older classes of antidepressants, such as tricyclic antidepressants and monoamine oxidase inhibitors. The mechanism by which these drugs act is usually not well understood. Treatment efficacy is only known after 6 to 12 weeks, and each drug carries a long list of side effects. So when an ineffective drug is prescribed, not only is it a waste of time for the patient whose symptoms do not improve, but there are also the side effects on other systems, sometimes causing long-lasting damage. Clearly, patients would benefit tremendously when these severe consequences from choosing the wrong treatment could be overcome. This underscores the need for biomarkers predictive of both remission and treatment failure, and is particularly relevant for anxiety and depression, as it is predicted that they will soon become the leading cause of disability. At present, there is no suitable biomarker for depression or anxiety, neither imaging-based nor otherwise. For anxiety and depression, the current literature of imaging biomarkers that target treatment response is highly heterogeneous, with generally underpowered sample sizes and a large amount of variation in techniques and analysis approaches. So the $1 million question is, how can we do better? And what is the crucial role of radiology, that is, of us? When it comes to the efficacy of psychotropic drugs, the saddest part is not that the effectiveness of treatment is so low, but that these numbers are not improving. The current efficacy is not better than 5 years ago, or 10 years ago, or 15. Studies fail to show how the care and treatment of mental health disorders has improved since 2005. This is particularly remarkable when we consider the incredible improvements in almost any other medical field. In the past 15 years or less, we have seen how infectious diseases have become less lethal due to response protocols against epidemics such as SARS and Ebola developed by WHO. We've seen how, thanks to the introduction of highly active antiretroviral therapy, HIV/AIDS has evolved into a serious but chronic disease with survival stretching into decades. And we are now witnessing how the concept of individualized care, also called personalized or precision medicine, is causing a revolution in cancer treatment. This approach heavily relies on biomarkers that take into account the individual genes, environment, and lifestyle. An algorithm weighing all these factors then guides the physician to the best medicine for that specific patient. We've seen none of this in the treatment of mental health disorders and desperately need more progress in this field. The good news, I believe it is possible. In the past decade, my research group studied the effects of psychotropic drugs on the development of the brain. One of our findings, common psychotropic drugs for the treatment of depression and ADHD cause lasting and maybe even permanent effects on the developing brain. For our research, we used imaging techniques on rats and humans. We also validated pharmacological MRI as a non-invasive alternative for measuring serotonin and dopamine functioning in the brain. This is based on the principle that drug challenges for these neurotransmitter systems evoke regional changes in neurovascular coupling, as well as changes in brain hemodynamics, such as cerebral blood flow, which we then measure. Such research is highly relevant since most of the drugs prescribed to children have never been tested in children before, only adults. The age-dependent effects we discovered are alarming because they imply that the most common psychotropic drugs prescribed to children, fluoxetine and methylphenidate, have been approved based on safety data which may be appropriate for adults, but not for children. We demonstrated the need for more drug safety monitoring studies on psychotropic drugs focused specifically on the treatment of children. This didn't make me very popular with pharmaceutical companies or with child and adolescent psychiatrists who now have to spend more time arguing with parents who are even more concerned about the long-term effects of psychotropic drugs. But the long-term effects may not necessarily be negative. Put simply, you don't want to prescribe ADHD medication to a child without ADHD, but for a child with ADHD, our data suggests positive effects on the brain, at least on the short term. We are currently analyzing data on the long-term effects, and I hope to present them in the next year. Although we have not yet established the predictive value of pharmacological MRI for the treatment response of mental health disorders, I believe pharmacological MRI has great potential as a non-invasive imaging biomarker. And by providing a more complete picture of effects and side effects, the treatment of patients with mental health disorders will evidently be improved. Studies like ours are not frequently conducted. Before we started, we wondered why, but after a decade of crashed clinical trials, a lot of research money, and 3 generations of PhD students, we know. These pharmacological studies are inherently complex due to the many assessments that are been done, such as 2 MR scans before and after the challenge, usually also a SPECT or PET scan and blood sampling, the many parties involved, the difficulty of finding medication-naive patients, the expensive scans and complicated data analysis. Yet we need more of it. I believe biomarkers could have the same impact on treatment of mental health disorders as they already do for the treatment of cancer. I believe that when it comes to the efficacy of psychotropic drugs, the numbers will be better in 5 years from now. They will even be better in 10 years from now. And in 15 years, they will be revolutionized. So, what are the most important challenges? Although some potential predictive imaging biomarkers have been identified, biomarker research in psychiatry is still in its infancy. In Nature Medicine, Rabi Dargan and Horga identified several challenges. To start with the most obvious one, for definitive psychiatric diagnosis, we lack a biological or histological test. The current diagnosis is based on a combination of symptoms alone, also called nosology, as reflected in diagnostic manuals such as DSM or ICD. The most common alternative for biological or histological tests, longitudinal follow-up, is imperfect. These follow-ups are often used to establish a final diagnosis and establish efficacy, but other factors, such as life events, may have diluted the association between nosology and the treatment response. Categorization of mental health disorders in DSM and ICD are often artificial and debatable. And also, pathological features of psychiatric disease seem to be very subtle and only appear when challenged. Finally, the costs of the scans are rather high. In the United States, MRI scans cost around $600 per hour in academic centers and up to $1,000 in commercial centers. And PET scans range from $3,000 to $5,000 per scan. In Europe, where I work, it is slightly cheaper, but still expensive. These challenges explain why biomarker research in psychiatry is still in its infancy. But I'm here to tell you that a new and emerging research field will change that: psycho-radiology. Now, let me be completely honest with you. The activities in this research field are not entirely new. Quite some talented researchers around the globe have studied imaging biomarkers in psychiatry before. Yet now we finally have a good name for this field. A field on the growing intersection between psychiatry and radiology that spans from diagnosis to treatment planning and monitoring. For me, this term symbolizes the shift in psychiatry from an era of subjective descriptive classification, the nosology I referred to earlier, into objective and tangible brain-based measures. All credits for the term psychoradiology should go to Professor Gong of Sichuan University. I was very happy to meet him last year at the previous edition of this conference. While preparing this talk, I again looked at his business card with many impressive functions. Maybe I would add one function, though, the one he introduced in a radiology paper 2 years ago: psycho-radiologist. So what is the current status of the field of psychoradiology? Psychoradiology has already identified several potential biomarkers, so far with moderate success. Cerebral blood flow in anterior cingulate gyrus, amygdala reactivity, and functional connectivity between frontal and limbic brain regions all seem connected to antidepressant treatment. However, some researchers found increases, while others found the exact opposite. But there is hope. A meta-analysis by Coil last year concluded that smaller hippocampal volumes predict non-response in depressed patients. Interestingly, volumetric changes to the hippocampus have also been strongly implicated in the etiology of depression. So hippocampal volumes could be a good biomarker when improving the understanding and treatment of depression. And I'm convinced that if we keep searching, we will find more predictive biomarkers. But in order to succeed, there is one thing we need: standardization. Let me give you 3 examples. Previous speakers already emphasized the need to standardize machines, acquisition schemes, and post-processing methods. I fully agree. Because of the extra challenges involved in biomarker development, this is even more pressing in the field of psychoradiology. These automated workflows focused on the quantitative assessment of imaging biomarkers will also induce a standardization of psychoradiological interpretations. For this, we need to set up large-scale consortia with a priori designed large-scale multi-site studies with coordinated analysis plans that include independent validation. This would mean a radical shift from the current post-hoc sharing of datasets acquired at multiple sites with differing goals. Expert consensus reports on clinical psychoradiological MRI examinations as was recently done by Professor Gong in China for patients with schizophrenia, could be a first step. In addition, we should create disease-specific dedicated working groups, for instance here at ISMRM or RSNA or neuropharmacological associations such as ACMP and ECMP. Replication using standardized paradigms and reporting in well-powered studies should be accepted as the norm. This means that the entire research field, including grant providers, should reward replication over novelty. It is striking and at the same time a bit embarrassing that although for more than a decade studies suggest that fMRI amygdala reactivity may be a suitable biomarker for antidepressant treatment response, only 2 years ago Nord and co-workers persuasively demonstrated that the reliability of this biomarker is too low for this goal. We need to do better. Finally, and most critically, to establish their clinical utility, biomarkers will need to be validated in representative, real-world populations. Inconsistent evidence on the usefulness of biomarkers for Alzheimer's disease has led to their exclusion from most evidence-based guidelines. Without consistency, clinicians might base the use of biomarkers on practical considerations, such as the resources and experience, rather than on evidence-based considerations. Later today in the evening session on the advances of psycho-radiology, I will elaborate on the need for non-invasive biomarkers and the lack of studies integrating all genetic, clinical and imaging data and what to do about it. But ask me my main 3 priorities for the field of psycho-radiology, and I tell you, standardization, standardization, standardization. The world is a better place thanks to the field of radiology. Since the first discovery of X-rays in 1895, the world has never been the same. Now, almost 125 years later, imaging is no longer a scarce resource. It is often the first step in diagnosis. Also, in my hospital, I've seen how image-guided strategies are replacing formal open surgical procedures for many diseases, such as treatment of stroke. Now is the time to shift our attention to mental health disorders. Every year, 1/5 of our population suffers from a mental health disorder. More than 1.5 billion people around the world. Let us bring that number down. And in order to succeed, there is one thing we need: standardization. And let me finally express a little desire. Next year, when I return to this conference, I will take an extra good look at your business cards. I will closely study the function titles, and I hope to see a lot of people with a strange function title, a title that until a few years ago, or maybe until today, no one had heard of: Psycho-radiologist. And with that, I would like to thank the 3 generations of PhD students who did all the hard work, the funders for the research money, and you for your attention.
Thank you. Thank you very much, Professor Renneman. It's a great talk. I think in sense of time, I think we'll have to close the session now. On behalf of my co-chair, Dr. Mustafa Bashir, I just want to thank all the 3 speakers for their great talk. And finally, I want to thank you all here for your patience and participations. So thank you very much. I'd like to close the session now, and I hope everyone enjoy the rest of the conference. Thank you. Do you want to introduce
Well, good morning, everybody. Don't go away. There's still more to see and witness. All right. So thank you very much for joining us for our special session on the scientific highlights of the 27th annual meeting of the ISMRM. My name is Scott Reeder from the University of Wisconsin, and it's my pleasure to be joined up here by Carla Miller from Oxford University. And we're going to get things kicked off with the next 25 minutes. This is a real treat where we have some very specially selected abstracts for a general audience of some of the best work and most exciting work that has been submitted to the Society, brought to you by the program committee. So without further ado, we'll get started.
Wonderful. Okay, so the first talk of this session is titled First in Human Trial of Gadolinium-Based Theranostic Nanoparticles, MRI Assessment of Uptake and Biodistribution in Patients with Multiple Brain Metastases. And I believe it will be given by Dr. Dufour.
Thank you. So this is my disclosure. I'm a consultant for the NHTRX company. So let me first thank the Society for giving me the opportunity to present this work in front of such a large and distinguished audience. I will first present the theranostic nanodrug AGX that has been used in this clinical trial. It is shown here. So you have in the center a polysiloxane core on which are grafted on average 10 gadolinium chelate, DOTA. The mean diameter of the nanoparticle is 3 nanometers. And the reactivity is about 3 times larger than the one you can get from standard clinical contrast agent due to the larger size of the nanoparticle. It has also very nice specific tumor targeting properties. This is shown here on this rat brain image with a tumor. And beside, of course, these nanoparticles are therapeutic, so it's radiosensitizing nanoparticles, as is shown here. So in the presence of high-energy photons, for example, from radiotherapy, you have production of Auger electron, photoelectron from the electronic cloud of the gadolinium. Then these electrons propagate to the neighbors, gadolinium atoms, and they create secondary electrons, which in turn end up with free radicals and oxidative species that are deleterious for the cells and for the tumor cells if you are inside the tumor. So the NanoRad clinical trial was based on the idea that these nanoparticles could effectively enhance the efficacy of the radiation therapy in tumors. And it was designed with first, the objective to evaluate the tolerance and the best dose that could be administered of these nanoparticles intravenously in combination with whole brain radiation therapy. So we included here 15 patients with multiple brain metastases from 4 different types of primary cancer, so lung cancer, breast, melanoma, and colon cancer. And we had 5 increasing doses of nanoparticles that were injected to the patients, so 3 patients per dose. This is the timeline for the patients. So 2 hours after the injection of the nanoparticles, the patient went to the MRI room for a first MRI session. And 4 hours after the injection, he underwent the first session of radiotherapy. So in my talk, I will focus more on the MRI finding about this clinical trial, especially the tumor targeting and the measurements of the concentration of the nanoparticle in the metastases. So here you have different images for the 4 different types of brain metastases that are shown. T1 weighted images before and after the administration of the nanoparticles. And in order to better appreciate the tumor targeting of nanoparticles, here you have the signal enhancement map. So they nicely show that there is a nanoparticle uptake in the metastases. We observed this MRI enhancement for all types of metastases, all patients, and at all doses administered. If we compare the MRI enhancement that is obtained with nanoparticles to the MRI enhancement obtained with clinical contrast agent like DOTARM, for example, we find fairly similar values. especially if we take into account that the administration was performed 2 hours after the MRI session was performed 2 hours after the injection. And I have to say that this were obtained for the same amount of injected gadolinium for these both cases here. We observed a linear increase of the signal enhancement of our particles compared to the dose injected. And very interestingly, we have MRI enhancement 1 week after the administration in the metastasis, which is of course very nice for us for the radiotherapy purpose. Then we quantified the nanoparticle concentration in the brain metastasis. For this, we generate T1 map before and after the administration. Here you have an example of T1 map obtained after the administration, so you can see the 2 metastases here that have nanoparticle uptake. From this, we computed the concentration of the nanoparticle using this standard equation here. And we found out that the concentration in metastasis was ranging typically between 20 and 60 milligram per liter in the metastasis. Very interestingly, also, we had with a linear correlation between the signal enhancement that was obtained with our MRI sequence, T1 weighted sequence, and the concentration of the nanoparticles, as shown here in 1 patient. So I came to the, I want to summarize the results and come to a conclusion and give you some perspective about this work. So this is the first clinical trial with intravenous injection of gadolinium-based nanoparticles. We found out that the nanoparticles are very effective for enhancing the 4 different types of brain metastases. The enhancement are comparable to the one that could be obtained with that are obtained with a clinically approved contrast agent. We still have an enhancement 1 week after the administration, and also we're able to evaluate the concentration of the nanoparticles, which is important in view of the radiotherapy. And this can be facilitated by the fact that we have a linear correlation between the concentration and the MRI enhancement. And this is beyond the topic of my presentation, but we have also good tolerance to the injection, and the radiotherapy results are very promising. So we launched in March this year NanoRad2, which is a multicentric clinical trial for 100 patients. And on this, I just want to say merci beaucoup pour votre attention. Thank you.
All right. Thank you very much. We'll move on to the next speaker, who will be discussing objective lung density quantification in neonatal bronchopulmonary dysplasia via ultra-short echo time MRI with comparison to clinical severity and reader scoring. Dr. Nara Hagano.
Well, thank you very much for that introduction. It's really an honor to share with you all today some of our group's work using ultra-short echo time MRI to look at lung density in preterm infants. I have nothing to declare. So infants who are born preterm will often face a severe and chronic lung disease of prematurity, and this is what we call bronchopulmonary dysplasia, or BPD. But unfortunately, we really don't have a very good understanding of how the underlying clinical pathologies relate to later outcomes. So we think that there's real potential here for imaging to play a significant role. But historically, tomographic imaging has been relatively scarce in this population, due in part to their small size and in part due to their vulnerability to ionizing radiation. Fortunately, the field of lung MRI has made a lot of great progress in recent years, in particular with radial ultra-short echo time, or UTE MRI sequences. So our group has been able to apply this in the neonatal population. So we've shown that lung MRI has clinical relevance with regard to predicting clinical outcomes when we use a pre-existing reader scoring system. But of course, we know that reader scoring can be time-consuming, and it has some degree of subjectivity. We've also demonstrated that we acquire the UTE MRI in a proton density regime. So we know that we can treat the neonatal lung density quantitatively with good comparison to CT. So the overall motivation for this study was really to compare the quantitative metrics we're getting from pulmonary UTE MRI to these validated reader scores in this neonatal preterm population. So we recruited 55 neonates from our intensive care unit. Most of them were born preterm. All of them were imaged near term equivalent age. And we do this with a minimal additional risk to the patient. At our site, we use a neonatal size 1.5T scanner, which is sited within the NICU, but a conventional scanner would also do the trick. And for our 3D radial UTE pulmonary sequence, we achieve a CT-like 3D isotropic resolution. And also, we're acquiring our images in a proton density regime. So we get a lung density metric that is akin to grams per cc after you normalize to muscle intensity. So for our reader scoring, we use a pre-existing system called the OCI scoring system, and it's got various different categories that evaluate parenchymal abnormalities, and they're all scored 0 to 2, and I'll come back and revisit some of these momentarily. For the quantification, we're interested in the whole lung, so we segment every single voxel from our UTE images, and then we're able to plot a whole lung density distribution. So for a control patient like this, it might look Gaussian, centered on about 0.5 grams per cc. We can then quantify this distribution. In this case, we've chosen to do density bins, where we have the low-density emphysematous phenotype in blue, the high-density fibrotic and consolidated phenotype in red. A patient with BPD might look something a little bit different, perhaps still Gaussian, but more spread into those abnormal density bins. A patient also diagnosed with severe BPD might look even more exaggerated into those abnormal density bins with something akin to a bimodal distribution. And finally, a third patient, identical diagnosis, might look extremely different, a complete lack of emphysema, but a strong component of that fibrotic consolidated phenotype. So really, these density distributions can act as a bit of a unique identifier for the disease phenotype that each patient has. So our metrics of interest here include the percent of the total lung in each density bin, and then from our analysis, we can get a bonus metric, which we call hyperinflation, which is just the total lung volume normalized to the size of the patient. So when we compare, if we first start with the percent of the lung that is both in the low and high density bins, if you compare that to the score for subjective impression in our cohort of 55 patients, you see that this trends together relatively well. Also trending well together is the percent of the lung at high density compared to the scores for fibrotic opacity. And again, you get decent trends when you compare the quantitative hyperinflation to our scores for hyperexpansion. One comparison that doesn't go as well is the percent of the lung at low density compared to scores for emphysema, whether that's size of regions, number, or size and number of regions, which is what's plotted here from 0 to 4. And I think the reason that this one didn't go as well is actually due to a weakness in the reader scoring system that we used. So for example, a patient that has 2 regions of emphysema that barely exceed 5 millimeters in size, that patient receives a 4 out of 4 for emphysema, which is the same score a patient gets if they have emphysematous regions globally throughout the entire lung. And those are certainly not equivalent cases. So in this particular scoring, we think that, or in this particular evaluation, we think that the quantification actually does much better than the reader scoring system. So we're excited about this work because it means that we can objectively quantify these parenchymal density abnormalities in this preterm population using pulmonary UTE MRI. And we can do this with good correlation to reader scores, and we can do this with minimal risk to the patients. In the future, we're hoping to be able to use some of the rich regional information we can get from these images in order to improve disease phenotyping. We're hoping to show that the quantification is related to predicting clinical outcomes, just like the reader scores were. And we think that this type of method will also be able to be used serially in order to see whether or not different therapies are having an effect. And really, all of this is leading toward the goal of making individualized clinical decisions in this little understood population, driven in large part by the imaging itself. So I want to thank my colleagues, both at Cincinnati Children's and at University of Wisconsin-Madison, and thank you all very much for your attention.
Thanks very much. The next presentation is going to be titled Longitudinal Multimodal MRI Monitoring of a Novel Combination Therapy Using Radiosurgery and Checkpoint Inhibitor Immunotherapy in Glioblastoma Multiforme. And it's going to be given by, Dr. Duong.
Thank you, Karen. Thank you, Scott. My student couldn't make it here. So this is the project that we started with by a group of radiation oncologists and oncologists, medical oncologists. So we studied this in a mouse model. As you know, GBM has a very aggressive clinical course, and it has very poor prognosis. There are three major treatments, radiation, chemo, and immunotherapy, which is more recent. One of the things that are somewhat hot in recent years, is this so-called checkpoint inhibitor. And that's coming from Dr. Liu in our group and Stenson from our group. But basically what it is an immune checkpoint inhibitor is a type of drug that blocks certain protein made by the immune cells. This protein keeps the immune response in check and can keep T cells from killing cancer. So the proteins, if you block that, you basically put the brake on the system, and this checkpoint inhibitor cells would start attacking the cancer cell. So basically, it's a good way to have a very targeted immunotherapy. Recently, Dr. Liu and others have shown that the combination of immunotherapy and high-dose radiation induces abscopal effects that could be more effective than either of them alone in minimizing tumor infiltrations and also slowing down tumor growth. So what we want to do is to see if we can test this hypothesis, that is the combinations of immune and radiation, a single-dose radiation, to see if it slows down tumor growth and lengthen survival compared to no treatment, immunotherapy alone, or radiation alone. And this is tested in a mouse model of GBM. Very briefly, this is the model. That many cells, roughly in 7 in each group, 4 groups, non-treated group, the checkpoint inhibitor group, the radiation group, and the combined treatment group. This is done on 70, 15 days, 30 days after tumor implantations. We measure T2 volume, T2 ADCFA, and others. We also measure survival curve, and immunohistological analysis is formed at endpoints, which is roughly about 60 days. Here's an example of some of the images, the control meaning no treatment. You can see the tumors on this side at 15 days and different days here, 30 days. Radiation treatment generally have a small volume. The combination oftentimes usually have a smallest volume in terms of tumor growth. So here's the tumor. They all start roughly at around this tumor volume, and that's expected because this is before any treatments. So we treated at 14 days and 12 days and 14 days twice. And you can see the volume on 30 days down here. So what we found was that statistically significant in volume between the non-treated group compared to the radiation-treated group and also the combination therapy. We measure the T2. This is a whole tumor RI. We can do regional, but we started out with the whole RI. This is relative to the contralateral side control or normal T2. So this is expressing the changes. As you can see here, that at 15 days, which is in the green bar, you can see that the tumor volume, the T2 is higher than control. And there is not statistically different across different treatment groups. Okay? They're all bigger than control, but not statistically different between treatment groups. At 30 days, they're trending toward a higher T2 in the combination group, and we interpret that potentially as a edema where cells dies and there's more fluid into the system. We look at FA, again at 15 days, which is at the green bar. There's no differences in FA even between normals groups, and also there's no difference in the FA among different treatment groups. At 30 days, the FA of the combined group is close to normal, whereas the individual treatment group, which is the radiation and the non-treated group, has a higher FA compared. So this suggests maybe there's some normalization, although it's hard to tell from just MRI alone. So we also measure ADC and insulin in figure, but the conclusions, the results are generally the same. That is that there are changes in ADC, they're heterogeneous in the images, and you can, as you know, they reflect cellularity. Again, this is a whole tumor RI. We haven't done regional yet because this is a small sample size. Look at kurtosis as well. So the general result is that there are some heterogeneity within the tumor, and they changes among different groups. We measure survival curves. And as you can see here, this is the control non-treated group. They died within 30 days or so. Whereas the combination treatment group has the longest survival, and the other two is somewhere in between. So here's the histology at the end of the course, which is typically either 30 days or 60 days. You can see here that the tumor indeed, as shown, is representative of what we see with the MR in vivo longitudinally. Basically, the results here suggest that there may be some synergetic effects when you combine immune checkpoint inhibitor and radiation. We also do histological analysis, again with 4 different groups. The findings are that I actually don't know how to interpret this, but they're very colorful and pretty. So I will just tell you what the results are. There's an increase in CD8 lymphocyte infiltration in group 1, which is control. And CD8 cells are abolished by RT, as you expect, because you radiate, you abolish the CD8 cells. And CD8 cells restore if you treat it in combination. So in the image that you saw above, the CD8 cells show morphological features that's suggestive of phagocytosis by tumor cells. So this finding essentially suggests that radiosurgery acts via the in situ immune modulation in the infiltrating GBM and possibly abolishing the ineffective lymphocytes and promoted infiltrations of good working tumor cells, if you will. Here's my conclusion. The combinations of radiation treatments and checkpoint inhibitor effectively reduce tumor volume burden and improving survival rate compared to non-treated group, radiation alone or immunotherapy alone. We see changes, heterogeneity within the region of the tumors, and histological shed some light on the possible action mechanism. Thank you.
Very good. Thank you very much. The next speaker will be Hyung-Hoon Lee, discussing intact metabolite spectrum mining by deep learning in proton magnetic resonance spectroscopy of the brain.
Thank you so much for introduction, and thank you for the opportunity to present our work. I have nothing to disclose. I'd like to talk about NMR first, which is a well-established method for metabolite quantification. The NMR spectrum has an excellent data qualities, and there is no baseline in there. On the other hand, an MRS spectrum suffers from low SNR, severe spectral overlap, and most of all, broad baseline. And as a result, metabolite quantification is very challenging compared to the NMR. To overcome this challenge, nonlinear least squares fitting is most commonly used in MRS. However, fitting performance is known to be subject to variability depending on the setting. Therefore, developing a more robust quantification method is a remaining issue. So, recalling the NMR, if we have spectrum like this, namely intact metabolite spectrum, we can achieve very accurate metabolite quantification by solving a simple inverse problem. But this is what we have in vivo. So our question is, how do we get this sort of nice intact metabolite spectrum from in vivo spectrum? Given the remarkable success of deep learning in diverse areas, including MRI, we use deep learning to solve our problem. That is, intact metabolite spectrum mining using deep learning. First, we built a total of 50,000 brain spectra for training of CNN. We include different levels of metabolite concentrations, baseline, and spectral quality. Then, we divided the spectra into 3 data sets. And then, we train our CNN by using the simulated brain spectra as an input and the intact metabolite spectrum as an output. Once we have output spectrum, metabolite quantification is achieved by solving a simple inverse problem. This slide shows the performance of our CNN on the simulated spectra. These are the CNN predicted results, and as you can see, there is only a small differences between the predicted and the ground truth spectra. And these are the quantification results by solving an inverse problem. And as you can see, we have almost no residual signals. These results altogether means our CNN clearly extract the intact metabolite spectra that are fully accounted for by the metabolite basis set. This is the summary of the metabolite quantification in terms of MAPE. despite the fact that our simulated spectra include much worse than typical in vivo. The error for the most of the metabolites were within 20%, especially GABA and glutathione, which are very hard to quantify in vivo. Our CNN achieved an error below to 20% for GABA and close to 10% for glutathione in the test set. Next, we evaluate our CNN using in vivo spectra. Note that our CNN never seen true in vivo spectra before. Nevertheless, the CNN nicely extract the intact metabolite spectra that are fully accounted for by the metabolite basis set. This graph shows the metabolite quantification by using our CNN and LC model. For some metabolites like this, they are mainly comparable to each other. But for other metabolites, the CNN result is closer to the known concentration ranges than the LC model results. These are the results shown in a previous slide, and the scan times was around 2 minutes. And these are the results from the same volunteers, but they took only 20 seconds to obtain. Despite the substantial difference in scan time, the predicted results are almost the same. Therefore, our CNN can be very robust against low SNR spectra. So, we looked a little more closely at the quantification variability against the different signal averages. For several metabolites like this, the result did not differ very much in both methods. But for other metabolites, the CNN is far more robustness compared to the LC model. These results altogether clearly support the feasibility of so many proton MRS of the brain. Finally, we investigated the response of our CNN to the spectra contaminated by strong oral residue or ghosting artifacts, which are added to this original spectrum. Of course, our CNN never seen such spectra before. Nevertheless, all of severe artifacts were almost completely suppressed. However, there were noticeable differences between the predicted results with and without the artifacts. So at this point, we think that our CNN needs more technical solutions to handle this sort of issues. So to conclude, the robust quantification performance of the proposed method made possible to so many proton MRS for the clinical studies. I'd like to special thanks to my PI and my colleagues, and thank you for attention.
Great, and the final talk of the session will be titled Liquid Metal and Stretchable Tubes, a Wearable 4-Channel Knee Array. And it's going to be presented by Dr. Port.
Thank you for the introduction, and good afternoon, everybody. I have nothing to declare. We all come in different shapes and sizes, and so do the patients that come to our MR centers. And yet what they find there is MR scanner equipment that's of fixed size. So let's take the example of a knee exam, and this knee call here is rigid and it doesn't adapt to the patient's anatomy. And this compromises sensitivity for some patients and comfort for others. Now imagine a coil that would overcome this trade-off by adapting to the patient. I believe such a coil would be stretchable and elastic and would conform very well to the body part being imaged. And this would offer good sensitivity and good patient comfort at the same time. Now such a wearable coil would also enable imaging of the flexion of joints. And while this is a very specific application, it could offer very interesting functional insights. The wearable coil could also be put on by the patient him or herself, just like a piece of clothing and even outside of the scanner room, improving workflow. But what's the technical challenge behind this concept? For this, let's have a quick look back at the conventional marker from before. Inside, we have these individual coil elements, and often these are made out of rigid copper. And while copper offers very high electrical conductivity for good imaging performance, it's not flexible or stretchable at all, which is really what we would need for a wearable coil. And in recent years, very nice work has demonstrated the feasibility of flexible coils by use of, for example, screen-printed conductive paste, multiple resonator conductors, and high-impedance coils. Stretchable coils have also been proposed even some years before, and their main difference to flexible coils is that this coil conductor here can be elongated. In this particular approach, a copper braid is used, which however requires textiles with high restoring forces. One more recent approach uses a stencil printed liquid metal on neoprene foam. And here, however, very thin metal layers have only achieved rather low electrical performance. And also the sealing of this exposed liquid metal is an open question. And in our work, we address these issues, building upon using this liquid metal as a conductor material. And this liquid metal, a gallium-indium alloy, is actually a fascinating material. It's liquid at room temperatures, it offers high electrical conductivity, and to the best of our knowledge, has low toxicity. Upon deformation, and that's interesting when you think about building a stretchable conductor, it can actually return back to its original configuration. And we use this material and we inject it into a stretchable silicon tube, and for sealing and electrical contact to the liquid metal, we use a copper insert and heat shrink tubing. And this way we arrive at a highly stretchable MR coil. But how well can such a coil perform electrically? For good imaging performance of a coil, you would like the coil conductor to have a resistance as low as possible. And while gallium-indium has high electrical conductivity, a conductor made out of this material will still have a resistance that's 17 times larger than if the same conductor was made out of copper. And now this is for a frequency of 0 Hertz. Now luckily at a higher frequency, such as 128 megahertz, a phenomenon called the skin effect comes in, which forces the current to flow on an outer layer of this conductor. And this layer is 4 times larger for gallium-indium than it is for copper, which then effectively reduces the difference in resistance here to only a factor of 4 at 128 megahertz. And this nice effect we can also see in the measured unloaded quality factor, an indicator for the electrical performance of the coil, where here a value of 130 is achieved when the coil is not stretched, and also when it is stretched, still high quality factors are obtained. And at these values, imaging will be sample noise dominated, and we expect very little SNR degradation due to the resistance of the coil conductor. We then combine several of these stretchable coil elements into a wearable coil array. And here in this first version, these coil elements are attached to a stretchable textile by sewing. But we then wanted to create an array that's even more wearable, and for this we used 2 layers of a stretchable textile. And these are now sewn on top of each other in a way that creates these textile casings, into which then the coil elements are inserted. And here you can see our first MR images taken with the proposed system of a volunteer's knee at different flexion angles. Upon flexion, the kneecap slides over the thigh bone, and also the cruciate ligaments, they rearrange to accommodate for the flexion of the joint. We then also performed a dynamic study of the knee, where the volunteer was moving his knee freely inside of the MR scanner. And again, you can see the kneecap and the cruciate ligaments, how they rearrange to accommodate for the flexion. In the future, such a wearable coil could be complemented with wearable detection, eliminating cabling to the array by on-body digitization and MR signal transmission via an optical link. Altogether, I believe wearable coil technology may take us a step forward towards MR systems that adapt to the patients and not the other way around. Thank you.
Well, thank you very much for your attention. Thank you to all the speakers. We're going to close the session, and thank you all. Enjoy the rest of the meeting.